Hello everyone, welcome to the Team Beat. I'm Lai Yang, a graduate student at Washington University in St. Louis and co-producer of the Pain Beat podcast with Dr. Rebecca Seal from University of Pittsburgh. In this episode, we gather a fantastic group of scientists and clinicians to discuss channelopathies, a group of disorders caused by ion channel dysfunction that lead to conditions such as epilepsy, cardiac arrhythmia, and most notably, chronic pain. Today, we are joined by Dr. Angelika Lampert from Aachen University in Germany, Dr. Tham from University of Washington and Seattle Children's Hospital, Dr. Candace Paulsen from Yale University, and our moderator, Dr. John Del Rosario from Washington University in St. Louis. Together, they will discuss everything about channelopathies, from the challenges of messy human genetics and environmental factors to the exciting promises of personalized medicine and new therapeutic strategies. Thank you for joining us, and I hope you enjoy.
Hello, everyone. I'm John Del Rosario, an instructor in the Department of Anesthesiology in the laboratory of Dr. Robert Giraud at Washington University in St. Louis. Channelopathies are diseases developed because of defects in ion channels caused by either genetics or acquired factors. We will discuss how these channelopathies impair ion channel function and how they affect certain patient populations. Without further ado, let's go around the table and ask the panelists to introduce themselves. Angelika.
Thanks very much. My name is Angelika Lampert. I'm a professor for neurophysiology here at Aachen University in Germany. And here I'm also the speaker of the Scientific Center for Neuropathic Pain. My lab studies channelopathies. We are also very much focused and interested in voltage-gated sodium channels and how they are affecting excitability of peripheral neurons. We also really like the structure of the channel, and so we are looking into the 3D structural modeling, how mutations are changing it, how mutations are changing the function, and how this is then changing the function of the nerves, leading to the clinics. We are using several techniques like electrophysiology, patch clamping, but also transcriptomics and the combination of both, patch-seq. We also use stem cells derived from the patients who are carrying mutations in order to get a better picture of what is happening in the cells of the patient. We are really excited about that, especially about the translational part of this type of research.
Thank you very much. We can continue with you, Siwan.
Hi, everyone. I'm an associate professor at the University of Washington in the Department of Anesthesiology and Pain Medicine. So I'm a physician scientist, and my clinical training is in pediatric anesthesiology and pain medicine. In my research work, I am in the Pediatric Pain and Sleep Innovation Lab at Seattle Children's Research Institute, the Center for Child Health, Behavior, and Development. Now, this lab is really unique because we are a multiple PI lab. So broadly, we study pain in children. We look at the biopsychosocial factors that affect pain, the mechanisms that drive the relationship between sleep and pain. And we work on developing innovative treatments for managing both acute and chronic pain across various populations. So developing digital technology based on psychological therapies to be delivered to children with chronic pain, sickle cell disease, functional abdominal pain, and pancreatitis as examples. In my own work, I'm very interested in underlying mechanisms that really determine how sleep problems lead to pain chronification in children both without chronic pain and with chronic pain. And another area that we've been really looking at is understanding the different phenotypes in pediatric chronic pain so we can start to look at personalization of treatment.
Thank you very much. Candy?
I'm Candace Paulsen. I'm an assistant professor in molecular biophysics and biochemistry and the Wu Tsai Institute at Yale University in the United States. I am a basic scientist. My background's in chemical biology. And what my lab is really interested in is understanding diverse regulatory mechanisms of members of the large transient receptor potential or TRP ion channel family. And our initial work has been focused on the wasabi receptor, TRPA1, and different mechanisms that maintain normal function of this ion channel, and then how those mechanisms can go awry leading to the establishment of chronic pain. And actually, our first discovery we made in my lab was uncovering this unexpected mechanism by which a really drastic truncation mutant of TRPA1 confers gain of function. And that was a pretty fun story to figure out.
Great. Thank you. Let's continue about understanding what channelopathies are. The first open question is, how is the research of pain channelopathy going? What do you think is happening currently? And what is the most vulnerable population that you actually can see?
So I think it's still an extremely exciting field of research. We do have these really hard mutations, which are leading to very clear inherited diseases where you can have the whole families with generations of patients being affected by it. And that opens up this fantastic possibility to look into the mechanism to understand the role of these single ion channels in the whole context. And then we also have this other side, and more of this is coming up, where we have patients, and I think Siwan will probably go into detail there much more, where we do get genetics examinations, but in the end, we get a lot of unknown variants. Quite often, the human geneticist here comes to me and says, again, we've got so many unknown variants, and clinicians say, what do we do? The patient is really sick. Can you find a reason for it? And then we do our patch clamp magic and all these things. And then in the end, sometimes we can say, yes, we find something, but quite often it's complicated. And I think this is a big challenge now of the channelopathy field, because now we start to find more and more factors that may modify things and that may put more flavors to the hardcore mutations. We will have more and more regulatory genes that come into play. Maybe not only genes, but also environmental factors, social factors, other factors that heavily can affect pain. And this is what I really like about it, because now we start to put these things into context and to see what's the broader picture of these channels. How do they function in context? Yeah, and I think this is where we are in the moment.
In the case that you normally see one, can you explain a little bit more about what you encounter with those children that you treat?
Oh, absolutely. I'll take one specific clinical population, children with erythromelalgia. As you all are aware, there's primary and secondary erythromelalgia, and a portion of primary erythromelalgia, the etiology has been attributed to the sodium channel 1.7. At Seattle Children's Hospital, because we serve 5 states all around us, we have, interestingly, a fairly decent amount of children that we see for EM. Now, over the years, we have a proportion who have been shown to have a mutation, but actually the vast majority do not have mutations. You know, it's like the rule of medicine. You break everything down to a third, right? A third seems to respond to some form of medication, a third fluctuates, and a third do not. What was really interesting to me, and this is really anecdotal and just based on observations because the numbers are so small, that those with sodium channelopathy, they don't seem to respond to mexiletine, which is what we think is the most available at that time. Now, I know there's a lot of movement, for example, sodium channel 1.8 targets. But with mexiletine, they do not seem to respond well. And that's always been a real question for me. Why is it that a proportion of my patients who do not have demonstrated mutation in sodium channel 1.7 seem to be more responsive, but not the ones who have mutation? I'll just talk a little bit about the longitudinal course. If you look at the textbooks, it does not seem to indicate that there's significant resolution in folks with erythromelalgia, but over time, for a third of them, they seem to have complete resolution. Then I go back to questioning my diagnosis. Is it another pain diagnosis that could have similar presentations, but that is not as well reported because of the reduction in prevalence, because this is a rare disease?
I feel that it's kind of difficult to understand how each patient population is specifically modulated by all these factors. This is a very open question. Do you think that there is a specific environmental factors that contribute to that specific effect independently of the gain of function mutation? Will patients with gain of function mutations exposed to specifically environmental factors have a more drastic, more pronounced effect into their symptoms that they experience? Or how do you think that might be going on?
So for me, this is really one of the key questions. And it's also why we are taking stem cells off the patients and differentiate them. And we now started to do this on a larger scale. We are now doing a differentiation of like 15 patients in parallel, which takes a lot of time and effort. And we have to fight with a large variability because the method is just very new and you have to really look for all the different things that can go wrong. You have to make really good quality controls and all that. But I think this is the way where we can now actually try to find out what are the genetics contributions that are behind it, because you do in the stem cells that you take from the patients, of course, you have all the genetic variants in there. You not only have the hard mutations, but all the SNPs and other things that make me look how I look and you look how you look, which is, of course, obviously different. You can capture it in the cells. Then you can use these cells in order to, for one, understand the pathomechanism a little bit better. These experiments are still ongoing, but we now already see that you can distinguish even certain pathologies, the clinical diagnosis in the dish. And once you have this, then you can start to break it back down to what is expressed in these cells. And then we do electrophysiology on these cells, then we collect them, and then we check what is the transcriptomic identity of this specific cell. We really make it on the single cell. And then you can see, why is this cell firing more than the other? What is the genetic makeup?
Okay.
And then in the lab, you can also start to try to mimic what is happening to the patient. And this is why I think it's so important that clinicians and basic scientists work together very closely. And I really like when Siwan says, I'm super interested in the mechanism, because this is where we can start. Because then you start to look at the patients in a way that helps the basic scientist to ask the right questions. In the lab, we can start, for example, now we do temperature changes. This may be obvious in erythromelalgia because you can trigger pain by changing the temperature. But we also started to change glucose in the medium because some patients say that what they eat is affecting their health or their feeling of the pain. Or also trying to change the pH in order to mimic other stress-related changes potentially. Also, what we are now doing is screen of medication. And there, of course, these newly developed ion channel blockers, especially for sodium channels, they offer a new way of modifying. Maybe one day we will be there, hopefully not too far of a future, that we can actually find specific changes that need to be modified per patient. And with the new tools, then we can repurpose medication in order to help the patient specifically. But that only works if clinicians and basic scientists work together.
So you mentioned that you see differences and then you do the sequencing analysis to figure out what's different. Is that with cells from a single patient you have variability, or are you looking across patients?
So we do both. With iPS-derived sensory neurons, you quite often have the problem that differentiations vary so much that you have in two differentiations already, you have large variability. But nevertheless, you can control it. And within one differentiation way, you get a mix of different sensory neurons. And not all of these sensory neurons that you get are hyperexcitable. Some of them are not. Some are spontaneously active. Some you need to trigger. And so we are doing this patch-seq approach also in the iPS-derived sensory neurons, of one patient. And then we not yet comparing with the others because this is slow throughput. And we are trying to look at the TRPA1 as well, because I do think this is one of the candidates that need to be looked at. And this one is tricky to catch in the iPS-derived sensory neurons. What system are you using?
We do heterologous cells. I think what your lab does with the patient-derived cells is the dream. Echoing what Angelika said, you have to have a good relationship with clinicians to even get access to samples. So that is a post-tenure dream for me. But now what we've been doing is any modifications that either naturally exist or that we make in the lab, we will put them into heterologous systems. So either HEK cells or Xenopus oocytes. I guess we're the other extreme of the completely isolated system, which means that we're only going to be able to capture things where the biophysical properties of the channel are disrupted. And fortunately, I think TRPA1 is still newer to the scene of the channelopathy front, that there's been room for discovery. But I think there are tons of mutations that are identified or rare variants that seem to be very impactful to patients, but maybe don't have a significant impact on the biophysics of the channel. And I think that's where the more native context would be absolutely critical.
To me, TRPA1 seems always a little bit enigmatic because you can activate it with so many things, and then it's still hard to activate it. It seems to be somehow on a crossroad of all kinds of different regulation pathways. Do you have a handle on activating it? And can you maybe explain to me what is it that it is sensing?
So the way that I explain TRPA1 is it's our cellular fire alarm system for reactive chemicals. There are so many enzymes that have cysteines at their active sites. And if you have an electrophile floating around, that could modify those kinases or phosphatases and disrupt their activity. And that would cause whole cell tissue damage akin to high temperature, low pH. So we've evolved to have a protein that is exquisitely sensitive for these types of chemicals in order to initiate protective responses. As a chemical biologist, I love this channel. That's how I think about it. But I think for hardcore ion channel people, that is really frustrating because normally these channels are not this promiscuous, but that is actually the point of this channel.
I wanted to ask something, Candy. For the audience in the podcast, could you please describe a little bit more of the things that you have observed in your own research about TRPA1 channelopathy?
Sure. There are two gain-of-function channelopathic mutations that have been identified and characterized to date. The first one is a point mutation in what's called the S4S5 linker. It's a loop that connects the voltage sensor-like domain to the pore of the channel. And this is a mutation that turns an asparagine into a serine. And those amino acids are going to have different hydrogen bond networks. And something about that leads to channel hyperactivity. And that variant is functional on its own, and it's 5-fold more active than the normal wild-type channel. The second gain-of-function mutation is a drastic truncation mutant. So this is a mutation that introduces a premature stop codon at part of the pore, and it removes the last 201 amino acids. And on paper, that mutation makes absolutely no sense because the key piece that makes an ion channel is missing. And so it makes absolutely no sense that this thing would be gain of function, let alone functional. And what we found was that in isolation, which is usually how these mutations are studied, sure enough, that mutant is completely functionally silent. But if you co-express it with the normal wild-type protein, which matches patient genotype actually, then you get gain of function. So it turns out you need some complement of wild-type and truncated subunits coming together into tetramers. These channels assemble as tetramers. And something about that combination of subunits leads to hyperactivity. As you said, this is a very difficult channel to work with. This has been a very difficult hypothesis to continue studying, but I have a really good student working on it. We're trying to figure out how many wild type, how many mutant subunits are in these hyperactive channels, what they actually look like, and what's altered.
So the expression of TRPA1 is mostly in the sensory neurons, but is there other areas that might be also compromised as a result of this gain-of-function scenario?
TRPA1 is also in the keratinocytes. It is in the lung epithelial cells. So it's not exclusively in peripheral sensory neurons. And certainly the production of this specific variant could and likely does have effects across the board.
Angelika, could you describe a little bit more about the gain-of-function mutations on your publications?
I mean, it depends very much on the sodium channel you're looking
okay.
at. Let's stay with NAV1.7. So obviously there are the loss of function variants in NAV1.7. And if you do not have a functional NAV1.7, you have congenital insensitivity to pain. But what is really surprising is if you have a gain of function, a very strong gain of function mutation in NAV1.9, which is the slower brother, if you want so, of NAV1.7, you can also get loss of pain. So you have a gain of function in the ion channel, but you have loss of pain. And this is very typical for NAV1.9. And there people are thinking that this is due to a overexcitability of the cell, and thereby it inactivates all the other sodium channels in these cells, and therefore they cannot fire anymore. These patients are also affected with other side effects like gastrointestinal problems, and interestingly enough, they also have a hypermobility of their joints, which I find is an interesting link between sodium channels and proprioception. So this is the activation mostly affected, or like the channel doesn't function anymore. And if you now go to erythromelalgia that Siwen also mentioned, there you have a much easier activation of the channel. And then it's open and it fires, and these patients get these really bad effects. Then there's this PPD, which then affects fast inactivation. In these past years, we saw a couple of mutations of that, and you could see that fast inactivation is badly affected. And when you then look at the structure and you see the different mutations at the different places in the structure, it helped a lot to understand fast inactivation mechanisms much better. Now people have shown that some of the drugs are even binding in the fast inactivation binding site and things like this.
To that, I want to piggyback in what you were saying about TRPA1. What I'm wondering is something about what C1 and Angelika was saying, how these patients might be sensitive to eating a specific type of food, or they might be sensitive to a specific external stimuli. So in the case of TRPA1, which is a chemical sensor and is very promiscuous, that does not go from one side to another, in your repertoire of things that you activate TRPA1 with, Candy, do you include new compounds that are potentially part of diets of children's or any human that had say that maybe eating this make me more sensible to have this kind of behavior? Have you ever thought about including something like that in your research?
We've certainly thought about it. We have not done it yet. And what's really interesting is that for the two known TRPA1 channelopathies, one of the triggers is actually fasting. So the opposite of eating. That would suggest that there's a metabolic byproduct that starts to circulate, and that is what's triggering.
What about in sodium channels?
Yes, so the sodium channel is easily to be modified by small molecules. Mostly it blocks it. I think there are lots of metabolic substances that were just simply not tested yet that most likely are modifying the sodium channel. And now, of course, with the microbiome, you can start now to think about all kinds of metabolic things that are flooding the body from time to time with astonishingly high concentrations that most likely are modifying the sodium channel. So yes, I would be super interested to see how that is affecting the sodium channel and then the patient. And I mean, in a way, we also have these electrophysiological methods, which we don't only use in the lab, but you can also apply to the patients. In humans, of course, you can have microneurography. In adults, you can see directly how the nerve fibers are responding. This is a little bit more tricky to do in kids. In kids, you can do QST, like quantitative sensory testing, or there's also new electrodes or electrical stimulations developed. It's a lab in Denmark, and also now here a colleague of mine in Aachen, who are now designing pulse shapes that are very specifically activating sodium channel subtypes. And that is something that less invasive. You just have an electrode to put on your skin. And like this, you can have a highly translational way to look at the changes that are underlying in the nerve fibers of the humans off the sodium channels, and then you can follow it up in the lab. And then that is also something where you can much easier then also see how conditions like environmental factors also are affecting the physiology of the human, and then hopefully also the physiology of the ion channels in the lab.
I must admit, I've had so many questions in the last 5 minutes of this discussion. So I'm going to try and pick one question, but I'll come back to some of them. As someone who do not have background training in ion channelopathies or these low structures in my mind that usually far from my brain when I'm treating patients, is there developmental changes in expression of channels based on, I'm seeing huge nods.
Big time.
Over time, and what other implications to potentially early intervention in the context of symptom presentation or without symptom presentation?
For the sodium channels, we know that there is, during the development, quite some change. NAV 1.2 and 1.3 are exchanged to NAV 1.6. And also that there are splice variants that are expressed at different time points during development. And some of the known erythromelalgia mutations and other sodium channel mutations are more harmful when they are expressed in one of the splice variants. And this can also be one of the explanations why patients have pain during a certain period of time during the life, and then it is alleviated. So this definitely is changing. And then you were talking about the loss of function patients. And there, of course, it is still discussed whether they would not have some kind of counter-regulation. They seem to have still action potentials in their peripheral nerves. I mean, they can have flare on their skin, which suggests that there must be some kind of action potential, at least in the skin nerves. Sodium channels are there. Of course, we've got 9 different subtypes to choose from. Probably there's some kind of counter-regulation. I also think that if you have hyperfunction of them, most likely this is also changing the expression pattern. But this is not really well known.
What about the TRP channels and changes over the developmental lifespan?
The TRPA1 channelopathies, or the natural variants, that can have effects, or you'll see patients where those have effects from childhood to adult. So it seems like those variants can have functional consequences throughout life.
And from your clinical view, do you have the impression that also mutations where people are pretty sure that they are pathogenic, that they are somehow, the clinical picture is changing in the way you described it in the beginning, that it's disappearing, that the patient is coping with the mutation and doesn't have pain anymore?
Absolutely. I'm thinking of a couple patients where one patient especially, this was a very vivid memory of mine. I was walking through the wards at the hospital and I just hear this continuous screaming. It just went on for hours. I asked the nursing staff, what is happening? They said this patient came in with pain. Her parents said that she hasn't stopped screaming for 3 weeks. She was 11 years old at that time, and nobody knows what to do with her. For whatever reason, I work in the pain service, but we weren't consulted because the picture was so dramatic that it was hard for teams to conceive what was going on in the first place. And because erythromelalgia isn't as well known, so the diagnosis is not at the top of folks' head. But when I saw her, she was a clear case of bilateral lower limb erythromelalgia. We sent her to the ICU, started her on a lidocaine infusion. And within 6 hours, the parents said she got out of bed, went to the shower, took a shower, went back to bed. It was the most dramatic response I have ever seen. And so I transitioned her over to mixilotine. Luckily, it was helpful. And over time, we've been slowly tapering down mixilotine because symptoms just seemed to recede. And it was unclear because during the summer when she was in a sporting event, symptoms would go up and it will fluctuate over time. But finally, we just thought, okay, let's just stop and see what happens. And 6 years later, no more symptoms. It's hard for me to understand what happens. Is it developmental? I don't think it is a factor of coping and psychological changes. There's definitely that component of it because pain creates stress and fear and anxiety, etc. But there is something, I think, physiologically happening. And I don't know how to explain that with absolute certainty.
Yeah, there's a patient that we are investigating with Chip together with colleagues from China. He's Chinese. And he had also severe form of Chip with broken bones, with no pain. But later on during adolescence, he regained some of his pain sensitivity, which is also hard to explain. In the moment we are looking at two splice variants and regulation of genes and stuff like this. And maybe we find something there. We're still working on it. But I also think with the PPD, paroxysmal extreme pain disorder, the kids are suffering really badly of it. I think they are learning how to avoid the stimuli that are triggering these responses. And so they are somehow getting much better later on in life. So I do think there's also a behavioral and psychological component to it, where people somehow manage to find a way to deal with it. It's extremely hard to say what it is, and it would be so nice to know because then we could help so many people.
I agree. And you can see those in family clusters. We have a couple of family clusters where you have 3 siblings. They have the same genetic mutation. They grew up in the same household. They have, presumably the same environmental factors, but their expression of pain are quite different. This is particularly prominent. We have 3 siblings, the youngest still very little, but starting to show some discoloration and changes in the lower limbs. But the brother and sister who are about 1.5 years apart, one has significant pain-related disability, and one continues to be able to manage, life activities. When there's a physical examination of their symptoms based on just visual inspection, the symptoms appear to be similar. As you know, pain is biopsychosocial, and there are many factors that drive it. Mayo Clinic has reported on a series of folks go through intensive pain rehab program, And there were some improvement in folks who had undergone the couple of weeks in pain rehab program. And those programs are very intensive in providing multidisciplinary therapies, physical therapy, occupational therapy, psychological therapy. So I think you are right. I'm always envious of scientists where you're like, okay, we're just going to knock out this little bit and then we can test the effect on the other. And people are so messy.
Which is also the beauty about people.
Yes. So you had spoken to taking samples from patients and being able to identify the changes and mutation. How viable is that in direct clinical application? And what is the time for all of those steps to happen?
I mean, our goal is to really make it applicable in clinics. We are not there yet, definitely not. I mean, we shouldn't make wrong hopes here. With the setting that we have in the lab, we are now testing on these 15 participants. We are testing about 60 up to 80 different drugs that are already licensed. And now we do find some hits are nice because these were medication that were shown in clinics already to show therapeutic effects. So we can reproduce some of the clinic in the lab. I mean, we always have to think it's an IPS, so stem cell derived sensory neuron. It's on its own on our dish. We do not have all the supporting cells. We do not have all the environment that it normally has in the human body. But nevertheless, it's enough to at least see some of the changes already. And we need to improve the differentiation method and the readouts. And in the moment, we are differentiating for 70 days, which is a long time. But we are also working to get these cells into high throughput patch clamp machines in order to have single cell readouts. And then I do think that this could be a road where we then have the possibility to actually have this personalized approach. And now we have these variants, and we always focus on pain. And when you talk to the clinicians, and probably CYN had a similar experience, we realized that it's not only the pain which can be really impairing in the patient, but it's also the autonomic system which is affected. In one patient who had a very strong gain of function variant, she suffered from erythromelalgia, but she also suffered from cardiac arrest. She needed to have a cardiac pacemaker implanted, which shows that NaV1.7 maybe it is expressed in the autonomic system. We know that, but mostly focused on the pain. So maybe we should broaden our view here.
No, that's really interesting that you bring this up. And I'm struggling with lots of different conflicting thoughts and feelings. Having worked in the ambulatory pain clinics over almost like 2 decades now, it seems like the features that we ascribe to autonomic dysfunction have been increasingly reported. Years ago, maybe there's a smattering. But now it almost seems like every other patient who comes in will describe having palpitations. They often already have seen cardiology, got their echocardiogram. They have dizziness. They have presyncopal episodes, blurry vision, GI symptoms, primarily nausea. Many people in our area have constipation symptoms. And I don't know that we have a really good explanation of the comorbid autonomic dysfunction, and patients seek explanation. So what we have is a multidisciplinary treatment. So let me go back one step. The way that we try to conceptualize for now, and maybe it will change in the next 5 years, is that the autonomic dysfunction, we believe, is due to an imbalance of our parasympathetic and sympathetic systems. And therefore, we have to rebalance this. And what we have in our toolkit right now, they're not great. We may have a couple of medications, the beta blockers, for example, physical therapies with pacing and gradual increase in activity levels. And those are really what we can offer to patients. But to think of it in the context of a channelopathy, contributing to this is interesting. I don't know that I've conceptualized it as part of the etiology, but I would love to hear thoughts on that.
I mean, NaV1.7 is strongly expressed in the sympathetic neurons.
Mm-hmm.
These neurons are lacking NaV1.8, and thus they cannot deal so well with depolarization, and the whole function is a little bit different of these cells. And so if you have a gain of function in these neurons, you may induce activity. There's a really nice study from the Waxman lab from years ago. And there they showed that one of these arithmological mutations would reduce sympathetic activity.
I guess I'm struggling with how do you explain the temporal trends over decades, and how do you explain the changes over time? So for example, if patients participate in rehabilitation, their symptoms get better over the course of weeks to months. But having said that, I think about our obvious pain population, because pain is usually invisible, but there are a couple of conditions where pain is visible. And the other condition is complex regional pain syndrome. And so we have kids who come in with remarkable symptoms with discoloration and allodynia. And after 2 to 3 weeks of intensive rehabilitation, their symptoms resolve. And maybe it's completely different, but how do we explain channelopathies in that context?
I'm not sure whether CGRP is really channelopathy associated.
Yep.
I think there was a study trying to find variants in NAV1.7 and CGRP, complex regional pain syndrome, and they didn't really find an association. No. Not in 1.7, but it could still be in others. So I think we should not think of it exclusively. It could be contributing, but most likely there are also conditions where it's simply not a channelopathy. But if it's a channelopathy, I mean, we still have the problem with the trigger events, or of the events that you have the symptoms and then you don't. And we don't really understand it. So most likely there has to be something that then induces some kind of phase transition, that then the excitability in these neurons is completely changed for a while, until somehow the energy levels are changing back and the cells is coming back to normal. And this is something that happens obviously in the nociceptors, but it could, in my view, also very easily happen in the autonomic system as well.
Maybe on a related point, as a basic scientist, one thing that we really appreciate about channelopathic mutations is that it allows us to uncover other aspects of regulation of the channel. And I would say our biggest discovery to date, the channelopathic mutation led us to that, to the discovery of this other regulation pathway. But that regulation mechanism is intrinsic to the channel. It's definitely disrupted in the channelopathy, but it's absolutely critical for the wild type channel to function properly. And so what you're seeing in the clinic, or what's been changing in the clinic, could be modifications of many different types that is changing the activity or the environment of the channel independent of a mutation. By studying these mutations, that can lead us to a broader understanding of channel regulation, with the goal towards identifying therapeutic avenues.
I just figured out why my struggle. My struggle is I see pediatric pain as a treatable condition. Kids get better. This is what gets me out of bed every day. And so the idea of a mutation being a fixed outcome distresses me a little bit. And I think that I struggle with patients having this label. Yes, you have a mutation, therefore this is you for life. And whereas I'm always trying to, increase quality of life, getting them to their goals, especially with pediatric population. But the idea that it is the environment that can change the mutation is hopeful.
Yes, exactly. Following up on what Candy was saying, is that potentially in some cases, a channel doesn't actually have to have a channelopathy. Some of the triggers within the body could potentially modify specific intrinsic properties of these channels, which can mimic some of the functions that happens during a channelopathy. And that could resolve once the environmental conditions or the environment in which the channel has resolved. And I think that also plays a very essential role into that. With that thought, I want to wrap up with one specific question. How do you see the channelopathy field going? What kind of therapies should we be thinking about in the next 10 years? What kind of research should we be doing in order to specifically target modulation of ion channels? How can basic scientists collaborate more with clinicians in order to bring solutions to all of these different diseases and conditions that arise as a result of channel impairment?
I do think that we should use this breadth of new methods that are available now, like single-cell transcriptomics. I think we should improve our electrophysiological methods as well. Use new stimulation protocols. We are using those that developed like 30 years ago. and see whether we can use the new artificial intelligence approaches in order to understand better what is happening in the nerve fibers, in the little fluctuations, etc. And then see the channel in context. And I really like what Wen just said, that the kid is not the mutation, and the mutation is not the disease by itself, but it's just one factor. And it is a factor that may help us understand the pathology a little bit better, that may give us a target that we can treat, but it's still one factor in the whole disease. And there are so many other factors within. And I think the next step will be to understand the more complete picture, also with the big data methods that we have now.
I just want a little machine that tells me when I see a patient whether there are any channelopathies I can address, and then put in the context of the big picture.
Yeah, that would be fantastic, wouldn't it?
I'm curious about your thoughts on our latest FDA-approved sodium channel 1.8 suzetrigine. What are your thoughts?
It's fantastic. I mean, it's really opening up a new avenue, a new type of pain treatment. I mean, it's not as effective as I would have hoped it to be, but it's also kind of interesting, isn't it? We were always talking about NAV 1.7 and all the mutations in NAV 1.7. For decades, people were hoping that an NAV 1.7 blocker would be the solution. And now it's an NAV 1.8 blocker, which is interesting. Yeah, that also shows us that maybe 1.7 is one player in the whole system, and it may trigger then NAV 1.8, which is the bottleneck, and in a very decisive position within the system. Or maybe it's simply easier to get there for the drug to actually bind at 1.8 instead of 1.7. I'm absolutely sure that this is something that will develop really in an exciting direction, and that one day we will have maybe more specific blocker or a blocker that blocks 1.7 and 1.8, more combinations that will let us on. And maybe one day TRPA1 will be part of that game as well.
Is there anything for TRPA1 in the horizon?
Not necessarily that I'm aware of. I know that there's been attempts, like there are antagonists for the channel, but they don't tend to fare too well in the clinical trials. And so I think my lab's motivation of studying the full channel and the full landscape of regulatory mechanisms is to try to find other targets within the protein for therapeutic intervention. Right now, the drugs all target the really grease ball transmembrane domain, and I think that's what makes the drugs not particularly effective in the clinic, or why they fail in the clinical trials. And so we're trying to think outside of that box to find new avenues.
With that, I want to thank every one of you. Thank you so much for this insightful conversation. I truly appreciate all your expertise in having this amazing way of describing channelopathies and how this impacted the life of your patients and also your research independently. Thank you, Angelika, C1, and Candy for joining us today. We hope that this podcast is going to open new researchers coming in, new clinicians helping us be able to understand the landscape of many different ion channelopathies that are impairing the lifestyle of many people in the United States and around the world as well.
Thank you everyone for the inspiring conversation, and thank you for tuning in and listening today. We hope you enjoyed this fantastic discussion. This episode was recorded online on January 22nd, 2026 and edited by Lai Yang and Junie Dong. You can listen to other episodes of the Pain Bay podcast on Spotify, Apple Podcasts, and the Pain Research Forum.